One of the most extraordinary achievements of the last century must be the Green Revolution, resulting in a huge increase of food crop productivity. Modern farming techniques and crop breeding contributed to reducing undernutrition and improving living conditions for many. However, we're starting to see the real cost this revolution has had on societies and on the environment. The main production methods rely heavily on fertilizers, which are energy intensive to produce and environmentally damaging. Plants take up nutrients through the roots, but since the very beginning, most of them have evolved with a powerful support system, arbuscular mycorrhizal fungi. The extensive hyphal networks they form are intimately connected to the roots and extend far into the soil. Fungi are expert foragers, providing plants with water and nutrients in exchange for different carbohydrates. Everyone wins in this mutualistic symbiosis. However, it can happen that all accessible nutrients are consumed, and the remaining ones are locked up in a chemically inaccessible form. For example, phosphorus, one of the main essential nutrients, is abundant, but in a form that cannot be taken up by hyphae or plant roots directly. Special enzymes are necessary to make it available to them, and the current state of knowledge is that fungi cannot produce these enzymes at all. This is where we need to consider the complexity of symbiotic interactions happening in the soil and shift our focus to the most diverse group of all, bacteria. Some bacteria are capable of producing the required enzymes in large quantities, transforming inaccessible phosphorus into the accessible form, to be taken up by fungi and partly passed on to the plant. This functional trait some bacteria have could be used as an alternative to fertilizers, improving the uptake of already present and abundant nutrient reserves. In our project, we aim to better understand how and if fungi control their microbiome to favor these useful bacteria. This is not an easy task, because much remains unknown about the complexity of below-ground interactions, especially between fungi and bacteria. So, how do we go about this? We have designed a series of experiments and analyses we can do in the lab, based around the plants wheat and flax, 2 species of fungi and many potential candidate bacteria. We will filter through the bacteria in a screening process based on specific traits that interest us. This way, we hope to find useful bacteria that would make inaccessible phosphorus available to the plant by being closely associated with fungal hyphae. In the long run, these plant growth promoting bacteria could be used in the field as bioinoculants instead of fertilizers. The major challenge of the Green Revolution 2.0 is that the agricultural sector needs to maintain high yields but transition towards a lower input and more sustainable production system. And considering the crop, along with its complex microbiome, holds a potential that we are only just beginning to understand.